All of you come with different amounts of experience in this topic. So for those of you for whom this is sort of basic, please, I give you an apology. And for those for whom I'm going too fast, just feel free to ask questions during some of the panel sessions. So, genetics used to be sort of relegated to sort of this specialized testing that you needed extra sort of thought about. And now, as we've become more familiar with it, and as we've learned the limitations of our clinical evaluations without genetics, it's now been integrated into a standard diagnostic evaluation for people with low blood counts for which there's not an obvious answer. And particularly with chronic neutropenia, this has become part of evaluation. We recognize that one of the major barriers to this has been insurance approval. We can talk about that during the panel session, but we're working very hard through the registry, and I also chair the American Society of Hematology Precision Medicine subcommittee, and we're writing a white paper on this to try to help with insurance approval. So there's a lot of work going on to try to acknowledge that we need to educate our colleagues in the insurance companies as to the importance of this. So these are the major topics that we're going to talk about. Time is short, so I'm probably going to cut out the care coordination section of this talk because I know that we have a very sensitive time. The kids are coming back at noon, so I'm going to cut that part out, and we can just talk about that during the panel sessions. So as Dr. Neuberger mentioned, there's sort of different buckets into which we can consider the causes of neutropenia. In one cause, the neutrophil production is actually normal. Your bone marrows work fine. It's just something bad has happened to it, right? Be it medications, infections, autoimmune disease, or something else. And if you can just treat that other thing, the neutrophils can recover, right? And you can your underlying bone marrow function is intrinsically appropriate. Then there are other situations where the genes, and I'll talk about genes in a moment, that we have that specify that function to tell your bone marrow to make neutrophils may not be functioning quite right, and that intrinsically your bone marrow is having trouble producing neutrophils. Then there's the immune inflammatory conditions, which in some cases do not affect the bone marrow. The bone marrow is fine, and if you just treat the inflammation, it's fine. And others, the inflammation is intrinsically linked to neutrophil production, and you have to treat that differently from some of these genetic intrinsic bone marrow problems. And then, as Dr. Neuberger said, the idiopathic, meaning we don't know what's causing it, and that's where we're putting a lot of our attention as well in the neutropenia registry. So as part of our, I'll talk here about genetics, and then we have some talks on autoimmune and neutropenia in a moment. So what is a gene? So our bodies, right, are made up of cells, and each of our cells, well, most of our cells contain genetic information, with a few exceptions. And this genetic information is encoded on our chromosomes that contain the genes that basically are the footprint, are the instruction manual to the cells to tell our cells how to make different proteins or other components of the cell. So they are what specify all aspects of what makes us us, our height, our hair color, our neutrophil counts. So the genes are the unit of information. And there are genes that instruct the body how to make neutrophils at various types of levels. There are also genes that regulate the immune system, and there are genes that regulate the inflammatory system. And when these are impaired in their function due to changes in the genes that we call mutations, we can see problems. So just a word about nomenclature that Dr. Neuberger mentioned, and there has been a tendency in the medical field to lump things together. I'll give you the example of Shwachman-Diamond syndrome, which before we had genetics, we defined it as a certain constellation of clinical findings. It turns out that there's lots of different genes that can cause Shwachman-Diamond syndrome clinical manifestations, but the different genetic types of Shwachman-Diamond syndrome are actually distinct. They're not all the same. And so we're moving away from this sort of lumping everything together based on certain hand-picked clinical features and specifying the genes, right? So SRP54, I don't like to call that Shwachman-Diamond syndrome. I call it SRP54-associated neutropenia, for example. And this has been important because it's not just semantics. If you give something a label, it automatically triggers doctors to treat everything the same way, to monitor the same way, and they won't tailor. So in this era of precision medicine, where we now have genes, we can tailor for a specific patient how to take care of them, how to treat them, and it's an exciting era that's been opened up by genetics. So as I said, in the olden days, which is actually not that long ago, we relied on clinical features, right? And we still do need to integrate the genetics with the clinical features. It's not all just genetics, but genetics is an important component of this. And why do we do that? Well, some people who actually have a condition, a genetic condition, may lack sort of the classical clinical findings, but still be susceptible to the same complications. And then some clinical findings can overlap between different syndromes, so you can actually end up with misdiagnoses based on clinical findings that can masquerade as something that's actually different. So you need the genetics to confirm your diagnosis. And this is where often the insurance companies put up a barrier, and you can fight that actually and be proactive. And we're happy to help your doctor with that. There are certain papers, publications you can point to, and certain verbiage you can use to help them understand the importance of this. So now we have to, as part of diagnostics, we include both. Now, what are some problems? So basically, the main one is insurance, but it's also, there's differences in medical standards. The medical standards can lag, and I was just talking to some of you, thank you for sharing your stories with us, that there is a need, particularly for rare diseases, for patients and families to be advocates and to be proactive. And often you may have a fantastic doctor, but they just don't know about this particular rare disease. There's lots of rare diseases I know nothing about. But what I do is I then partner with experts in that rare disease, and we work together. And that's what hopefully you can do, and we can talk about that during the panel discussion. How do you build your medical team so that your local, very excellent hematologist who may not know a lot about severe chronic neutropenia can then partner together with sort of an expert team to work together? Because you need sort of boots on the ground hematology to take care of the day-to-day, but then you need sort of higher level input to make sure that you're getting the best cutting edge treatments and medical care. So why do we care about genetic diagnosis? Why does it matter? I'll give you a few examples, and then I'll give you some cases just to illustrate this. So for example, some of these genetic neutropenia conditions carry an increased risk of cancer, typically leukemia. And not all of them, so don't panic, but some of them do. And it's different. The leukemias that we treat in those conditions are different from the leukemias that arise in someone who's otherwise healthy. So on the top, we have a bone marrow. On the left, sorry, I don't have a pointer. I don't think I have a pointer. Do I have a pointer? Yes, I do. Here, all the bone marrow cells carry that same gene that can predispose to leukemia. Here you have a healthy marrow that just happened to develop a rogue cell that developed into a leukemia cell. Here you have a rogue cell that developed into a leukemia cell, but if you treat the leukemia and get rid of the leukemic cells, here you're left with healthy bone marrow. Here, in somebody with a genetic neutropenia condition, all the other cells that are left still have a predisposition to go on and develop other leukemias, so you have to treat them differently. Also, some of these neutropenia conditions involve other organ systems, the digestive system, the bone system, the liver, the lungs. So knowing your diagnosis helps us be proactive to keep track of all the other things that we need to monitor and keep people healthy. The treatments, the standard treatments that we use to treat, for example, with a bone marrow transplant that my colleague Dr. Myers will talk about, or some of the cancer treatments, et cetera, can be toxic if we use the standard regimens that we use for everybody else. We have to sometimes tailor it appropriately to the specific genetic condition. And actually some of the therapies are also, I'll give you some examples of that, targeted. And then one of the most important reasons why you want to know your diagnosis is the best bone marrow donor for a patient is a matched sibling. But if you know the genetic diagnosis, you can test the sibling to make sure they don't actually have the same gene that doesn't work. Because if they do, you don't want to use that marrow because you'll just give the patient right back the same disease. And there are published cases where siblings who look for all the world fine were used as a donor, and then the patient actually developed the same disease again, even though the donor is looking okay. So you have to be careful, particularly before transplant. So what are some of the challenges? So genetic testing used to be very expensive. The costs have come way down. And so that's not usually the main barrier now, but sometimes insurance coverage can be. Time is against you. So genetic testing can take time. And sometimes it takes a lot of time to work through all the results. And if you wait until you really need that result, for example, you need a bone marrow transplant, if you wait till that time, you may not have the luxury of time. If someone develops leukemia and you need to treat that leukemia, you don't have the luxury of time for this kind of genetic diagnosis, and you just have to kind of take your best guess. So you don't want to wait. I've heard some people say, and even doctors say, my patient looks fine. Why should I bother with all this extra genetic testing? And the answer is you don't want to wait till you really need it because you may not have the opportunity to do it. And then this is probably more for physicians, but some families as well. Genetics is not perfect. Our knowledge is not perfect. And some people are worried about what we call the variant of unknown clinical significance, right? Where you get a result back and we're not exactly sure what it means. When that happens, it doesn't mean that it's sort of kind of important. It means it's either causing the disease or it's not causing the disease. We just don't know which it is. But we have strategies, and that's where you can reach out to experts. And the Neutropenia Registry is available to you as a resource to help you work through how do we tease out what a variant means. And then lastly, genetic testing is a personal choice. And so this should be done in partnership with your physicians. And we've often provided consultation through either the registry or people have come to various centers where there's expertise in these very rare disorders to partner with your local doctors to try to help you through this. So there's the other thing that I wanted to mention along the theme of being proactive is it's not a one and done thing. Unfortunately, the genetic testing is very complicated. And there's a lot of reasons for that. And we don't have to get into all the nitty gritty. I'm happy to do that if you want during the panel discussion. But it's a little bit different, for example, patients with solid tumors. Patients with solid tumors, they usually get a single blood test, one and done. Is it a genetic cause or not? And then they're finished. And to be honest, a lot of the data that our insurance companies rely on are based on data from the BRCA breast cancer, genetic studies. And there it's a one and done. And so they assume that that's the case for neutropenia conditions. That is not the case. Often sometimes you have to use not blood, but you have to test skin fibroblasts because the blood can be misleading. Sometimes the genetic panel that's used isn't perfect. There are a lot of panels out there called neutropenia panels, but none of them contain all the genes. There are constantly new genes coming out. So sometimes there's a mistaken assumption that if you do something called whole exome sequencing, where you sequence all the genes in theory, that should be the best test, right? It sounds like the best test. You're testing all the genes. There are certain technical limitations to whole exome sequencing where certain types of mutations can be missed. It's not as good as some of the panel testing. So and the problem is if you start with exomes, often genetic companies will not then let you go back to targeted testing, even though you try to explain. The targeted testing pick up stuff that's missed on exomes. That insurance companies will say, you've already tested all the genes, we're not going to pay for any more. So what we've typically done as a strategy is started with targeted sequencing because you really, really target and analyze the genes that you want. And then if we come up empty, then we'll go to exome. So we can help you through different strategies with your doctors if they have any questions. So that's the whole point of that. You don't need to know all the details, but the point there is you have to be proactive and don't feel shy about reaching out for expertise. So just a little bit about genetics to give us some background on how to talk about this. We all have two copies of almost every gene, right? One inherited from our mom, one inherited from our dad. So there's two copies, and there are different flavors of these genetic diseases. One is called dominant, where if only one of the two copies is not working, that's enough to cause disease, right? And there's another type of neutropenia conditions that are called recessive, where both copies have to not work right in order to cause disease. That's called recessive. And then there's something called X-linked, which we can see with, for example, Barth syndrome. I don't know if anybody from the Barth Syndrome Foundation is here, but we've been working towards improving, sort of strengthening our synergies with the Barth Syndrome Foundation. This is where boys have only one X chromosome, and this is a gene that's found on the X chromosome. So it tends to occur in boys because they only have one of them if that X chromosome carries a problem with the Barth syndrome gene. So that's called X-linked genetics. Why does this matter? Well, when you do genetic testing, we'll get to why that matters in a minute. So when you get your genetic testing results, as I said, it can either be likely causing the disease, likely totally fine, or we don't know which it is and you have to work through it. So why does this recessive, particularly for recessive diseases, and this is a very important point, often, and you'll notice in the SCNIR, we often ask for parental samples when we're trying to understand a patient and trying to figure out if there is a genetic cause for the neutropenia. And again, not all neutropenia is genetic. I don't want to give that impression. But if it is genetic, parental testing can help because there are two potential issues. If you identify two variants in a given gene, it could be that each copy of that gene carries a variant, and that would cause a recessive disease. However, it's also possible that one copy of the gene carries two variants. It's got two hits, right? But it doesn't matter because you still have one functional copy of the gene, and that is not sufficient to cause the disease. And the easy way to figure this out is to test parents. If it's this situation, you will find one mutation in one parent and the other mutation in the other. In this situation, you'll find both mutations in one parent, nothing in the other parent. And so that helps you figure out whether you found the diagnosis or not. So that can be very helpful. So I'm just going to give you, I'm going to end with a few illustrative examples because I want to save time for Dr. Neuberger, how genetics can inform treatment. So this is a little boy with a history of neutropenia and marrow hypocellularity, meaning the marrow is not as full of blood-producing cells as it ought to be. He had some GI issues. He wasn't absorbing nutrients normally. His immune system was abnormal. His immune system was impaired, and he couldn't make antibodies appropriately. And he had recurrent infections, including respiratory infections. I'm sorry, I used some medical jargon here. Respiratory infections, and he had skin infections, right, recurrently. And his initial clinical genetic testing was unrevealing. So they signed up for research testing, and we found through whole genome sequencing, a gene called PLCG2. So he was originally called Shwachman-Diamond syndrome because of this combination of GI sort of malabsorption of the gastrointestinal system. He couldn't absorb nutrients appropriately, and he had neutropenia. So that's usually Shwachman-Diamond syndrome, but he had PLCG2, and he had a pathogenic variant in that. Why does that matter? PLCG2 is a gene that causes something called PLAD, which is an immune dysregulatory condition. And what you do is if you target the immune system with anakinra, which blocks the sort of inflammatory immune hypersignaling, you can actually ameliorate those symptoms. And his GI symptoms went away, his neutrophils returned to normal, his infections went away. He used to have debilitating fatigue. He couldn't go to school many days because he was so tired. And now he's doing sports. So this is an example of how knowing the gene can really help you because he does not have Shwachman-Diamond syndrome. And he does not, by the way, have leukemia risk. In the past, the parents were worried. Does he need regular marrow exams? Now the answer is no, he does not. Here's another example. Here's a boy with a history of poor growth. You'll see a theme here because Dr. Myers and I run the Shwachman registry as well. We get a lot of referrals for Shwachman-Diamond syndrome. He was thought to have Shwachman-Diamond syndrome. He wasn't growing well, wasn't digesting nutrients properly. He had bone abnormalities. He had neutropenia. He was on GCSF, and some of his other blood counts were also low. He had immune abnormalities, lots of infections, and he also had some neurologic issues, which we can see sometimes with Shwachman-Diamond syndrome. And all the testing for Shwachman-Diamond syndrome and the clinical testing was negative. So then we went through whole genome sequencing, and this was actually through a collaboration with the Broad because these are very difficult mutations to detect. And so we worked with a specialist in mitochondrial diseases. The mitochondria are the power engines of the cells. They generate energy. And he had a mutation in a mitochondrial gene. And this is just, you don't have to pay attention to all the science. But this was very important because we treat mitochondrial disorders very differently. Barth syndrome, for example, is a mitochondrial disorder. And so he should not be treated like Shwachman-Diamond syndrome. And this was a very important aspect. And now we have to worry about metabolic issues. So that's just two examples, and we could go on and on. So what do you do if the genetic evaluation is negative? Well, one possibility is it's not genetic. Another possibility is it is genetic, but something's been missed. So that's where research can be helpful. We have way more powerful tools on the research side than on clinical lab testing because the tools are advancing so quickly. And frankly, our ability to analyze is advancing quickly. And I would urge the other important thing is to make sure that your genetic results are reanalyzed regularly, because we've learned more and more about how to do the analysis, and we've been able to identify the last patient with the mitochondrial disease was diagnosed over a decade after I started working on him. So we never give up, and we keep working on it, and we regularly reanalyze. But even clinically, just keep having things reanalyzed. And these are not just conditions of children. We're identifying these genetic conditions now in adults as well. So we're particularly interested in adults with sort of immune-mediated neutropenias. Okay, so this is the last issue. So you can inherit mutations, and those mutations are in all the cells of your body, but some mutations can be acquired specifically in the bone marrow cells, and these can cause secondary issues in the bone marrow. These are called somatic mutations, and we use these to track complications that can occur. I'm going to just go really quickly. If I were to give you an analogy, a healthy bone marrow I'm going to illustrate it with a car, can chug along normally and make neutrophils normally, right? It functions the way you should. What happens with an Elaine bone marrow is it gets a little stuck and it can't quite finish the journey to making a neutrophil. What happens with these mutations is sometimes the bone marrow acquires these mutations and it can kind of juice up the bone marrow and sometimes help those bone marrow cells to grow, but in sort of a bad way, so that the bone marrow cells kind of crash and burn and they develop leukemia. So that's bad. And what we're finding now also in the laboratory there are also genetic changes that can be acquired in the bone marrow that can actually compensate for the inherited genetic mutation and now restore bone marrow function. We're trying to use this therapeutically. So we're trying to develop medications in the laboratory that can help, based on data that we've learned through studying the normal changes that happen in the bone marrow of patients with neutropenia conditions. Can we target those therapeutically? And we're working very hard on that, and other people are as well. So we hope that through some of this work that we're doing, we'll be able to come up with treatments. So this is just a schematic to show you that we're trying to use these samples to understand the cause of neutropenia and to develop targeted treatments. I'm going to hand the podium over to Dr. Neuberger now and switch out the slides. I'm not going to go into care coordination. I just wanted to add, because Dr. Dale raised some really important points, and I just wanted to follow up on that because I had run out of time, that for these different patient-reported outcome studies where you share your perspectives and your experience, these are critically important not only for doctors to better understand how to better treat you, but actually, as we develop these therapies, one of the questions that we're asked to address is, does it help patients, right? How do we show that this is effective? And if we don't have a baseline of what patients are experiencing before the treatments, we don't have a comparison for after the treatment. So this is a very important sort of baseline study that will help us sort of set the bar, and it also helps attract the attention of pharmaceutical companies because they need to have metrics of success. And so by participating in the surveys, you'll be helping sort of set the bar that we need to then improve. So thanks so much, and I'm going to hand the podium now to Dr. Neuberger to discuss gene therapy.
So thank you. Dr. Shimomura mentioned a number of reasons to find a gene for a given condition, and it helps us understand the reason why the condition develops, and sometimes, as she mentioned, there may be a drug other than GCSF that more fundamentally addresses what's called targeted therapy to help alleviate the neutropenia and perhaps other parts of the condition. So the genetics is very important both for children and adults. Another reason is that there's a rapidly developing field of gene therapy, as several people have mentioned already. And so if you know the gene, there may be a way to either replace or edit that gene to take care of the condition fundamentally rather than treating it, you fix it. And I'm just going to give one example for Elaine neutropenia, which is the most common form of severe congenital neutropenia. This is going to get fairly technical. I'll try to explain it in lay terms. There will be a short quiz tomorrow morning. So Elaine is a gene that encodes a protein called neutrophil elastase, as Dr. Dale mentioned. And this particular, the mutations that cause neutropenia in Elaine actually produce a protein that either doesn't fold properly. Proteins are produced as a long chain of amino acids, and they need to fold properly to function normally, and they have to go to the right place in the cell. Elastase is a really nasty enzyme that digests other proteins, and if it's not packaged properly, it damages a cell. And it turns out that having neutrophil elastase is not necessary. Elaine expression is dispensable. And actually, I discovered this when I was reviewing somebody else's article for a medical journal, where they talked about a syndrome I had never heard of called Papillon-Lefèvre syndrome. And this is due to a defect in a protein called cathepsin C, which is necessary for the processing of other proteins. And so if there's a defect in cathepsin C, not only is there no neutrophil elastase, but there are a bunch of other proteins missing as well. But these patients are immunologically normal, except they have really bad gums. Also, there have been no reported cases of neutropenia due to loss of Elaine. It's only been bad protein being made. So we figured, well, maybe you don't need Elaine. And one of the easiest forms of gene therapy is what's called a knockout. You get rid of it rather than fixing it. So another reason for using a knockout is that it's hard to fix individual mutations when, for instance, Elaine has more than 200 reported mutations all over the gene. So it's better to have what's called a universal approach to gene therapy. Now, when we look at a gene, we describe it as having exons, 1, 2, 3, 4, 5 for Elaine. Sometimes there can be 2 or 3. Sometimes there can be scores of exons, and these are the building blocks of a gene. And what we found in analyzing the genetics is there were a lot of what are called missense mutations all over the gene, but all the frame shift mutations, which lead to what's called a nonsense codon, were out here. And the reason for that is that there's something called nonsense-mediated decay. Nature abhors a vacuum. Nature also abhors nonsense. And so when a gene has a defect in this part of the gene that produces a nonsense reading, there's a destruction of the messenger RNA that takes information from the nucleus to the cytoplasm. And so the gene is not expressed at all. And our thought was to trigger the nonsense-mediated decay to get rid of the gene product by introducing this kind of frameshift mutation in this part of the gene, get rid of it. So the strategy is to collect hematopoietic stem and progenitor cells, the cells used in a bone marrow transplant, collect them from a patient out of the body, meaning ex vivo, edit the gene by these new tools based on CRISPR-Cas9, which has been in the news for quite a few years. It was, I think, the molecule of the year cover of Time magazine a few years ago. And fix the gene in the test tube and then essentially do what's called an autologous bone marrow transplant with the patient's own cells. After conditioning the patient, put the cells back in, they would engraft, and you'd have fixed the patient's own cells. And as Dr. Shimomura mentioned, there are 2 copies of each gene. In Elaine neutropenia, 1 copy has the mutation, the other is wild type, normal. If you fix the mutated gene, you fix the disease. If you fix both of them, you fix the disease. There's no neutrophil elastase produced, and that's okay. So either way, you get neutrophils produced with or without elastase. And the object is to make normal neutrophil production without a need for GCSF, which should cure the neutropenia and decrease the risk of myelodysplasia or leukemia. So we've done this in the test tube with a number of patients' cells. This was a patient who had a specific Elaine mutation. And, oops. After introducing this early exon gene editing, the cells, which were previously only looking like early precursors of the bone marrow, started producing neutrophils and measured quantitatively this improved neutrophil production. And this was then done with a variety of different mutations in different patients, and it fixed all of them. So this has all been happening in the test tube. We've also done it in what are called xenograft mice, where these are immune deficient mice which are able to accept human stem cells and make neutrophils. They do not make neutrophils in stem cells from patients with congenital neutropenia. We edited them, and then the mice produced normal human neutrophils. So the next step is a very small step, just from mouse to man. And this is lots of things work in the mouse and don't necessarily work in man. So the clinical trials are necessary. Clinical trials are very expensive, but we're now working on trying to put together clinical trials which would demonstrate that this would be applicable to humans. It's been done in a number of other diseases, and we're very hopeful that within the next few years, it may be possible to bring it to human trials. So this is just one example. Elaine is probably the easiest target for congenital neutropenia, but the same approach can be used for many others. So thank you.